Doug Kerr, Stem Cell Research.
All right, well, hope everybody had a nice lunch. So, just like in the morning's talk where we did a primer on neuroanatomy and neuroscience, you're going to be a stem cell biologist by the time you get done with this talk and the one with Charles French Constant that comes after. So I will talk about some of the work that has gone on at Johns Hopkins while I was there and still is going on there, in part a collaboration back to Biogen, where I am now. And we'll look at a couple things in this talk. One will be your own stem cells and what they may do and how we can kind of harness the power of your own stem cells to repair the nervous system. And Charles will follow with some more discussion about those stem cells that you have. And then I'm going to turn to kind of stem cells as a therapeutic strategy and ways we can begin to use stem cells or think of them to repair the nervous system, a field which is still in its early days and which we have a lot more to learn about before we can really do this effectively. So here's the lesson. So a stem cell is a cell which basically doesn't have any unique features except that it can differentiate into a specific specialized cell type, and another daughter cell from that stem cell remains a stem cell. So stem cells can come from a variety of tissues, and their ability to turn into mature cell types that make up tissues in our body is what makes them special, and their ability to renew. That stem cell continues and can repair the tissue is also what makes it special. Here you're seeing a hematopoietic stem cell, if I can get the pointer, and here is another blood cell which is present in your body and has the ability to replace tissues at all times during our life. So stem cells can come from a variety of sources. One that we've heard an awful lot about is the embryonic stem cell, which comes from an in vitro fertilization for couples who cannot have a pregnancy the conventional way. A sperm and an oocyte are fertilized in an egg, and that then fertilized oocyte becomes ultimately a blastocyst. And what you're seeing here is a blastocyst. And this is smaller than a pinhead, but contains a series of cells called the inner cell mass in here, which are embryonic stem cells. And the power of embryonic stem cells is that they can now be taken out, expanded in a dish indefinitely, and can become virtually any cell type in the body. Cartilage cells, bone cells, muscle cells, cardiac cells, nerve cells. So they are very powerful cells. They can be grown and expanded indefinitely, potentially for therapeutic purposes. And the more near-term goal for any stem cell, including embryonic stem cells, is really to study disease, in that you can recreate critical features of a disease in a dish. And if we can recreate aspects of a disease in a dish, we can begin to identify targets that might be used as therapies for that disease. Stem cells can also come from fetal tissue. They can come from even adults. And so there are lots of sources for stem cells in general. The further on you get towards adult stem cells, the more restricted a stem cell is. That is, most stem cells that you get from an adult human are restricted to the tissue from which it is derived. So you can get blood stem cells that can turn into blood, but they can't really turn into neurons. Same thing with skeletal muscle. There are stem cells in our brains and spinal cord that are restricted to that lineage. So they're less powerful in a sense when compared against embryonic stem cells. But they may be actually easier in some senses to actually generate mature cell types. So this is what happens. From however you generated your stem cell, you've now got a cell which can be grown and expanded in a dish. And then the magic of it really is that you can take that undifferentiated stem cell, here an embryonic stem cell, and can differentiate it to each or a variety of different tissue types. Pancreatic cells, hematopoietic or blood cells, cardiomyocytes, neurons, or hepatocytes. Now, for a long period of time, that was a stochastic or random process. You couldn't really control which lineage you would get. You knew that these were stem cells because they made all of them. But what has really enabled biologists now is that you can specify and direct the differentiation of stem cells toward a specific lineage. You can make, in a dish, literally hundreds of millions of neurons, or hundreds of millions of cartilage cells, chondrocytes, things like that. So now you have the ability to generate large numbers of these cells that you can really study for disease, and ultimately, potentially for therapy, to transplant into somebody. So what could we use stem cells for? Well, the one that we all know and talk about is replaceable tissues and organs. So if our liver is damaged, or our kidney, or our brain, or our spinal cord, that we could actually use the stem cells to replace that damaged tissue. Now that's very complicated. I told you this morning that the nervous system is hundreds of millions of wires that are connected all together, going up to and down from the brain and spinal cord. It is impossible at this point to conceptualize reconnecting that with the use of stem cells. Now, that's not to say that they won't have therapeutic benefit, but the complexity of the nervous system certainly is too hard to replace completely by the use of stem cells. So there are other reasons why we would use stem cells. So there are defective cell types that we can replace, oligodendrocytes, for example, or in some genetic disorders, you don't make certain enzymes. And so the transplanted cells can provide that enzyme. And there are examples of that currently with genetic neurologic disorders that have been very effectively treated by stem cells by providing that replace of the defective cell type. We can also think about delivery of genetic therapies. In this sense, it's like a Trojan horse. The cell is provided to allow the gene therapy and the product of the gene therapy to be provided to the tissue in which it's needed. And even for cancers, people are developing stem cells to deliver cancer drugs right where you need them, right next to the tumor. So again, if you look at adult versus embryonic stem cells, they each have certain advantages. They each have certain disadvantages. So the embryonic stem cells are pluripotent. That means they can turn into anything, any tissue in the body. Whereas adult stem cells are multipotent. They can turn into several tissue types, but they're more restricted in what they can become. Embryonic stem cells are highly proliferative. You can make hundreds and millions or even billions of these cells. Whereas adult stem cells have a limited proliferative capacity. Embryonic stem cells are non-autologous. That means it's not from you. And so therefore there may be problems if we were to transplant those stem cells back into you because they're not genetically identical. Whereas adult stem cells can potentially be done in an autologous way. So somebody could have their own stem cells taken out, expanded, and ultimately transplanted back into you. No risk of infections, no risk of genetic rejection. There are ethical concerns that continue to swirl around embryonic stem cells, and because they are so powerful, one of the things that we have to continue to think about is they may be in some senses too powerful, because there is a potential that they could form tumors, whereas adult stem cells are non-tumorigenic. Now you may have heard about induced pluripotent stem cells. This is a way to make an embryonic stem cell-like cell, but from the skin. So this was really maybe the most important scientific finding of 2007 by several groups, in which you could take skin cells, throw in a couple factors that would de-differentiate those cells. In other words, it would take them from a mature skin cell back to a pluripotent stem cell. And so now you've got, and it's called an iPS cell, so it's human-induced pluripotent cell, that basically looks in many respects like an embryonic stem cell, except it's not from an in vitro fertilization, it's from your own skin. It's genetically identical to you, that you can then push down towards specific lineages like cartilage cells or nerve cells. Now, a lot has to be done to convince us that you can reliably get these cells in large numbers. But again, the near-term benefit of this is that you can really start to study diseases in a dish. So you can use these cells to generate neurons or oligodendrocytes. So when we think about which stem cells to use, we really have to think about what do we want them to do. And this is where I'm going to show you a little bit of data. Do we want the stem cell to immunomodulate? In other words, do we want the stem cells to rebalance the immune system? Because in autoimmune disorders, including transverse myelitis and MS, the immune system is out of whack. And remember, it's starting to attack your own tissues. And there's very good evidence, actually, that stem cells will do that. They will kind of reset the immune system back to a more balanced state. We may want them to neuroprotect. That is to kind of shore up the already damaged nervous system and keep those nerve cells alive, keep the axons firing. We may want them to remyelinate, and in fact we do. We would ask some stem cells to actually differentiate into oligodendrocytes and re-wrap around those axons or wires that we were talking about this morning. And depending on what your purpose is, you may use a different type of stem cell for each purpose. All right, so we are viewing stem cells in the context of inflammation because transverse myelitis is an inflammatory disorder, MS is an inflammatory disorder. So does that impact the stem cell itself? The answer is yes, and it's in a complicated way. In some senses, the inflammation hurts the ability of stem cells to repair the nervous system. In some senses, it's necessary to allow the stem cells to repair the nervous system. So we have to sort all this out, right, before we can really understand what these transplanted stem cells would do within the brain or spinal cord, is we have to figure out what's going on in the brain or spinal cord and be able to understand what effect that would have on the stem cells themselves. But we all have endogenous stem cells, and Charles will talk about this next. We all have stem cells in every tissue in our own body, including our brain. And in fact, we know that these stem cells are active. They repair damage to most of the tissues or organs in our body. But they don't really do a very good job in the nervous system. So why then? Why would we have stem cells in our brain and in our spinal cord, and yet when we have a stroke, or Parkinson's disease, or spinal cord injury, or transverse myelitis, they don't fix the damage? That's a very important question, because if we can understand that, and if we can increase the power of our own stem cells that exist within us at any moment, that would be an easy way to reduce repair. And we're starting to learn about this, and this field is really exploding. What I'm showing you is an example of what stem cells do in the nervous system in response to injury. This is actually an example of a stroke. And here you're seeing a bunch of blue dots, literally millions of little blue dots. Each of them is a stem cell. And in response to a stroke, on that side of the brain, the stem cells activated and began to proliferate exactly what you would want them to do. So they become much more in number on this side relative to the side that doesn't have an injury. Well, that's good, right? Because we want them to be able to repair the stroke. Well, as it turns out, not only do they do that, but they migrate specifically to the area of injury. That's good too. And then they start to differentiate into the cell types that were damaged in stroke, but also in other disorders. That's good too. And that's where things go wrong. Because at that point, they've activated, they've proliferated, they've migrated, they've gotten to the site, they turn into the cell types, and then they sit there like bumps on a log and do not actually repair. So understanding that is key. And if we can figure out how to harness their potential and enhance their ability to differentiate and repair, that would be a major leap. And we're getting there. So harnessing these endogenous pools is our ideal. Proliferation and migration of endogenous pools of progenitors or stem cells to sites of injury does occur, but differentiation of those cells and integration into the nervous system fails. And ultimately, with chronic disease, such as progressive multiple sclerosis, ultimately the stem cells kind of give up. They say, look, we've been trying this for 20 years, and it's just not working. and they become depleted. So ultimately you may need transplantation, but still there's a lot of room to go with understanding and harnessing the power of these endogenous stem cells. So how do we cause them to become active? How do we cause them to divide, to migrate to the injury, to cause them to differentiate, to integrate? I will tell you the bottom two bullets here. One of the things that we absolutely unequivocally know increases stem cells in your own brain is exercise. So exercise doesn't just make you feel better. Exercise actually triggers new neurons in your brain. So that's one of the reasons why exercise is really important. The other thing, stress and depression actually decreases the rate at which new neurons are born in your own brain. And so treating depression, getting stress under control, these types of things are not only important to how you function, but they're important to your brain structure and generating new neurons. We can talk about that later. Okay, so is there a therapy that's in development that might enhance the ability of endogenous stem cells to repair a central nervous system lesion? Yeah, there is actually. And this is one that is being developed at Biogen, which is where I work. So this is a program that is currently in humans, in MS patients. And the idea behind this therapy is that here is an oligodendrocyte precursor or stem cell. And it is looking for its axon. And what you'd like would be for an oligodendrocyte to remyelinate a demyelinated axon. But in the case of MS and transverse myelitis, I've already told you that those stem cells don't do the trick. They get there, but they don't differentiate. So what if you block a specific signaling pathway called LINGO? LINGO is a protein that's on the surface of the oligodendrocyte, and if you block it, you find a fairly remarkable thing happen. These stem cells look very different after you have blocked this specific signaling pathway. So this cell has lots of myelin now that is just ready to wrap around an axon. And in the context of demyelination, these then continue to differentiate and wrap their arms around these demyelinated axons. So the idea is that after a transverse myelitis or multiple sclerosis, you've had this halting of differentiation that you can now overcome. And you can force these stem cells to become fully differentiated and myelinate. So here's an example of it. I'm going to show you a couple, and don't worry about these words on the left. These are different models of demyelination. But what I would like you to look at are these grayish or blackish circles. And let me explain what that is. What you're looking at is 10 or 15 axons, these long wires we talked about, coming out of the board at you. Okay? These are the axons. And the gray circles here are actually myelin that's wrapped around that axon. And each oligodendrocyte wraps around it multiple times, creating this grayish or blackish circle. So that's a myelinated axon. Now, in this injury, look on the left, you don't see any of those big grayish, darkish circles, because there's no myelin. So those axons are there, but they're demyelinated. But if you look on the right, now you've remyelinated. No transplantation of stem cells, nothing like that. You just harness the ability of the endogenous pools of stem cells to do their thing. Same thing in another injury model here, and EAE is an animal model of MS, or transverse myelitis. And again, you see the axons coming out at you. This is on the bottom right, and you see these gray circles around each of the axons telling you that they have been remyelinated. So, again, that's in clinical trials in multiple sclerosis. We're hoping to look at this in transverse myelitis. Now I want to switch to transplanted stem cells, and where are we with this? I'm going to show you some data on a particular stem cell called a GRP, or a glial restricted precursor. These are stem cells that can be isolated from the brain or spinal cord and are quite limited in what they can become. They can only become glial cells, hence the name glial restricted precursors. What are the glial cells? Astrocytes and oligodendrocytes. So in the context of a demyelinating disorder, in which the oligodendrocytes are damaged, this is a cell type that we would very much like to transplant into that demyelinated lesion, such as the spinal cord, to allow for remyelination. And there's some beautiful work by Steve Goldman in the States that shows that these cells, glial-restricted precursors, can generate huge amounts of myelin in the context of a demyelinating injury. So this is just a genetic model of demyelination, and everywhere you see green in this microscopic picture are these axons that have been fully myelinated. Very impressive that they can get into the nervous system. And by the way, these are human glial-restricted precursors transplanted into an animal, and they work just beautifully. This is what it looks like on a really, really, really high power. So I told you about these wraps of myelin around the axon. All right, so here's back to our neuroanatomy lesson. This is an axon, and then each one of these is a layer of myelin induced by these transplanted stem cells. Here's an even higher power view, and it's so high power that you can't even see most of the axon. All you can see are these layers and layers and layers of myelin wrapping around that. So that's what these transplanted stem cells can do, in this case, in the spinal cord of an animal with a genetic injury. Okay, so what we wanted to do, and I'm going to show you a little bit of data, don't worry about this, except when we first started the Transverse Myelitis Center, we really wanted a model of transverse myelitis. So we created one in which there's this, poor little rats, we actually induce a transverse myelitis injury, and they get weak and they have bowel and bladder dysfunction. Because what we wanted to do was transplant then stem cells into these animals to see if we could trigger a repair or remyelination. And this is a spinal cord of an animal with transverse myelitis, and I won't bore you with the details, but this looks very much like human transverse myelitis. Each of the blue or purple dots here is an inflammatory cell that is coming into and attacking the spinal cord just as it does in human transverse myelitis. And it causes a couple things that are also found in human transverse myelitis. Let me direct your attention to the top right. This is a stain from myelin. And you would love to see here a beautiful sea blue color that would detect myelin, but it's all white because this animal's spinal cord is demyelinated, which is exactly what happens in transverse myelitis. In the bottom right here, you're actually looking at axons, because axons become damaged in transverse myelitis as well, and the silver stain identifies individual axons coming out from the board, and you see that there's an area in the spinal cord in which the axons have been injured, just as there is in transverse myelitis. So, okay, great. So we've got an animal model of transverse myelitis. It looks like human transverse myelitis. What happens when we put glial-restricted precursors or stem cells into that animal's spinal cord? Well, as it turns out, they survive, they proliferate, they divide, they encompass the entirety of the lesion, and then what? Then they sit there like bumps on a log, just like the endogenous stem cells, and decide that they're going to be lazy and not actually fully remyelinate. So we came up against the same wall that the people studying endogenous stem cells came up against. That is, we can't get them to fully go all the way. And they go most of the way, but it didn't help. So we've had to go back and start to think about ways in which we can push them a little bit further and see if we can get them to actually remyelinate. And this is where we brought in our old friend, Lingo. So we would actually take these stem cells prior to transplantation, and we would say, look, we're just going to put this Lingo therapy in there, just push them towards a more mature myelinating state, and then transplant them in. And what happens in this context? Well, they actually do a lot better. So there's a lot more myelin. What you're looking at here are microscopic images in which each one of the transplanted cells is green, and red is myelin. So the more red you see, the more myelin we've reintroduced. And you can't see it particularly well because it's bright here, but we don't have to show it, unless it's very easy to do.
No, it's the switches. The switches.
Yeah.
Switch them all off. There you go.
Perfect. So, if we look after 3 weeks after transplantation, we see a bunch of these cells, the transplanted cells. We see a little bit of red, which is the myelin. But as we go 7 weeks later, now we see the whole field myelinated. So we really have been able to trigger a remyelination of the damaged spinal cord in this animal model of transverse myelitis. And here's just another one in which you remember the earlier slide in which this whole area was demyelinated, and now there's an awful lot of myelin back in place. And it associates with the animals actually starting to get better. They walk a little bit better. They're not perfect. Even though the myelin is actually looks very good, the animals aren't perfect, but they clearly are better. Okay, so it is on this basis that we ultimately would like to do a trial in transverse myelitis, not with exogenous stem cells or transplanted stem cells, not yet, although that's in the works as well. But the first therapy would be to try to boost up the power of the endogenous stem cells that we've all got in us, and that may be by looking at this lingo-blocking therapy. And there are lots of reasons why transverse myelitis is attractive to develop therapies. One, there's only one lesion. Two, it's unlikely to come again. Three, it's easy to do a surgery, easier than it would be, say, into the brain. Four, there's a whole bunch of outcome measures, electrophysiologic tests and imaging tests that can allow you to answer the question as to whether or not you're really making a difference in this case. So we've made that argument for quite a while, and I think it's caught on both for the stem cell companies, but also for those who are developing remyelinating therapies, and I think exciting days are ahead. Now, there is a problem, though, in that we don't want to necessarily be going right into the spinal cord with our therapy, right? So sticking a needle into the spinal cord, maybe not too bad, but I'd rather not have to do it, right? So is there a way in which you can get the stem cells into the nervous system without directly putting them there? And there's another problem, because many people have lesions in their brain as well, or several lesions in the spinal cord. So how would we think about treating many or all of them? You can't put the stem cells into all of them. This is an MRI of a patient with multiple sclerosis, and you can see each of the white spots here is a different lesion. So we've got to figure out a way to actually deliver the stem cells there. Well, how about using the blood? How about taking the stem cell, giving it into the blood, so that it then goes into the nervous system, into the brain, into the spinal cord to trigger its repair. Let me show you a cartoon, I think. So here are stem cells that we gave into the blood, and that's pretty much what our first couple years of experiments showed, is that they didn't get into the brain at all. They just stayed in the blood, they just went right on by and into other organs. And that was just a couple years of failure. So we decided actually that we would genetically modify the stem cells so they would be sticky, so that they could potentially get into the nervous system. We thought we knew how to do this because we now know how immune cells get into the brain. We know that how they dock with the blood vessel and get into the brain. And so we figured we would trick the stem cell by expressing the same molecules on the surface of the stem cell and seeing if they would then migrate into the brain. So this is what it looked like. Now you're going to see our new stem cells in which we've got a sticky protein on their surface. And here's my experiment. This one you'll like better. See, now they don't go away. Now they bind to the surface of the blood vessel, especially in the context of inflammation. As it turns out, in demyelinating disorders, the blood vessel itself gets sticky and expresses this docking protein. So now the stem cells that express one and the membrane, the endothelium expressing the other, it's a lock and a key. So now they don't migrate on by, they stick right to the surface, and voila. Well, that's the cartoon version. Wish it were so easy. But it does seem to be the case. I'll just show you one piece of evidence of this. This is a paper which is about to come out. But you're looking at an MRI of an animal that was transplanted this way. And now you're seeing the brain that is reconstructed. from this animal, and now just the brain. And now what I'm going to show you are the stem cells that have migrated into the brain from the blood, and it's them. So each little pink dot, I don't know how well you can see it, and there are literally tens of thousands, is a stem cell which has migrated into the brain in various regions, and much of the brain of this particular animal is engrafted with stem cells, and as we rotate the brain around, you can see that there's a very widespread engraftment. More on the one side of the brain, say here, because we've given it into blood vessels on that side of the brain, but much of the brain is engrafted. So it gives us hope that even in diseases that have a large lesion or many lesions, or brain and spinal cord, that we can start to gain some traction in treating these disorders rather than just focusing on those with a very small lesion. And we'd rather, of course, not to put the stem cells right into the spinal cord. So I will stop there and take any questions.
I think with your agreement, we'll wait. But